Hello everyone, let's start with the star of molecular biology, DNA or deoxyribonucleic acid. DNA does not build protein directly. Instead, it sends messages through RNA, and that's where tRNA comes in. tRNA or transfer RNA is a small but crucial type of RNA involved in protein synthesis. It makes up about 10 to 20% of the total RNA in a cell. Here are the key facts. Its sedimentation coefficient is 3.8s. This reflects its size and mass during configuration. Composed of 73 to 93 nucleotides. Its structure includes a unique clover leaf shape which holds into an L shape in 3D. tRNA is synthesized in the nucleus of the cell. It transcribed from tRNA specific genes on the DNA template. Interestingly, only about 0.25% of the total DNA is responsible for coding all the tRNA, a small portion with a huge role. Let's break down the key parts of tRNA molecule. First, acceptor arm where the specific amino acid attached. Second, anticodon loop contains a sequence of 3 bases that are complementary to codon on the mRNA strand. Third, T-Cy-C arm that help in ribosome binding. D arm assist in tRNA recognition by amino-acyl tRNA synthetase. Variable loop varies slightly between different tRNA types. All these features help tRNA do one thing: match the correct amino acid to the correct codon on the mRNA. The function of tRNA. tRNA picks up a specific amino acid in the cytoplasm and carries it to the ribosome where an mRNA strand is waiting. The anticodon on tRNA pairs with the codon on mRNA, ensuring accuracy. The ribosome link the amino acid together to form a polypeptide chain, a protein. So tRNA acts like a molecular adapter, translating nucleic acid language into amino acid language. Without tRNA, the genetic code could be meaningless. It ensures the correct amino acid is added in the correct sequence, making every protein precise. From enzyme to hormone, muscle fibers to antibodies. None of it exists without this tiny RNA molecule doing its job. Let's quick recap. DNA's double helix stores genetic information. tRNA reads that code, binds to the matching mRNA codon and deliver amino acid to the ribosome. mRNA stands for messenger RNA, also called as template RNA. It was named by scientists Jacob and Monod in 1961 during their work on bacterial gene expression. To carry genetic instructions from DNA in the nucleus to ribosome in the cytoplasm where proteins are made. mRNA makes up only 3 to 5% of total RNA in a cell, but it is essential for the production of every protein in our body. It has a sedimentation coefficient of 8s, which refers to its size in centrifugation. mRNA is produced in the nucleus through transcription. The enzyme RNA polymerase binds to the DNA and reads the template strands. It builds a complementary RNA strand, initially called pre-mRNA. This strand is then processed. Introns are removed, exons are joined, a 5' cap and a poly A tail added. Once matured, The mRNA exists the nucleus, but it does not go alone. In the cytoplasm, mRNA often travels with protective proteins in a structure called informosomes. Informosomes are spherical complexes that protect mRNA from degradation and guide it to the ribosome, the protein factories of the cell. Let's look at the 7 structure components of a mature eukaryotic mRNA molecule. A modified guanine nucleotide at the start protect mRNA from enzymes and help in ribosome recognition. Non-coding regions 5' UTR, the untranslated regions before the start codons, important for mRNA stability and translation control. The initiation codon AUG, AUG code for methionine, signals the start of protein synthesis. And coding regions, mRNA contains the actual genetic message, each 3 base codon, 1 amino acid. Termination codons UAA, UAZ and UZA tells the ribosomes where to stop translation. The second non-coding region UTR are present at the 3' end after the stop codons. They help in mRNA degradation, timing and translation efficiency. The poly A tail 50 to 250 adenine bases at the 3' end present. They increase mRNA life and improve translation. Together, these parts help the mRNA survive, get translated and make proteins accurately and efficiently. So what does mRNA actually do? It carries the genetic code from DNA to ribosome, act as template for building proteins, works alongside tRNA and rRNA during translation. This is the heart of the central dogma of molecular biology: DNA to mRNA, mRNA to protein. If there is no mRNA, there is no proteins. And if there is no protein, cells cannot function. Ribosomal RNA is the RNA component of ribosomes, the protein factories of all the cells. It makes up 80% or more of the total RNA in a typical cell. Yes, it's by far the most abundant RNA. Unlike mRNA, which carries instructions, or tRNA, which brings amino acids, the rRNA builds the ribosomes itself, the platform where proteins are assembled. In eukaryotic cells, rRNA is synthesized in the nucleolus from a specific region of DNA called the NOR, nucleolar organizer region, on certain chromosomes. That's why the nucleolus appears dark and dense. It's busy making rRNA all the time. In prokaryotic cell, like bacteria, rRNA is made from special ribosomal DNA, rDNA, regions directly in the cytoplasm. So whether it is a simple bacterium or a human neuron, rRNA is essential for life. Let's break down the 3 main types of rRNA based on molecular weight and sedimentation coefficient. First, high molecular weight rRNA at more than 1 million molecular weight, found in the large ribosomal subunit, for example 28S rRNA in eukaryotes and 23S rRNA in prokaryotes. These are long molecules responsible for catalyzing peptide bond formation during protein synthesis. Second, medium molecular weight rRNA. The examples are 18S rRNA in eukaryotic small ribosomal subunit and 16S rRNA in prokaryotes. The functions, they help in mRNA alignment and ribosome subunit stability. Third, low molecular weight rRNA. They have approximately 40,000 molecular weight. For example, 5S rRNA found in the large subunit of ribosomes. It plays a role in ribosome assembly and stability. These S values like 18S, 28S stand for Svedberg units, which reflects how fast the molecule sediments during centrifugation, related to both the size and shape. So what exactly does rRNA do? It provides a structural framework for ribosomes. It helps bind mRNA and tRNA during translation, and most importantly, rRNA act as a ribozyme. Yes, rRNA catalyzes peptide bond formation between amino acids during protein synthesis. In fact, the 28S rRNA in eukaryotes or 23S in prokaryotes perform this function, not proteins. This makes rRNA one of the oldest and most important catalytic molecules in biology.